Ready? Okay. So Maddie, question: why is this baby in that machine? Okay, why? Hmm. So there is actually a movie, not a good movie about this. Somebody knows why this kid is here? Duncan. Polio? Well, Asher, Angus. Okay, so far you think that the baby's giving this to other people. What about if it's the opposite? Mhm. So yes, Julian. Yes, yes. So it doesn't have. So when you hear HIV, so human immunodeficiency, yeah. So this one is not that. This one is a severe immunodeficiency. So this one is even worse. So do you remember when we're talking about metaphorically about bubbles that you have to see other people, that you have to only be with in contact with few people of your family? This is actually a literal bubble because if she goes outside of that environment, she will die. Okay. Um, so this picture is from the 60s, and I'm going to tell you a little bit about membranes and vectors because this is one of those diseases that we have a lot of tools to actually work on this. Uh, um, Adriana, do you know what is gene therapy? Alicia, have you heard about gene therapy? What is gene therapy? That every time when they were talking about the vaccine, the RNA vaccines, they were talking, oh, this is just gene therapy. Do you know what exactly that means? Yeah, okay. So we're going to talk about that and that inserting a gene and put it in our somatic cells as this kid will need to survive and to be able to leave that bubble is incredibly complex. Yeah. So I'm going to show you how in a few years now we can, in many cases, we can help this person, yeah, this kid by manipulating DNA and creating new healthy immune system. Okay. So the beginning, I'm going to talk about what is biohacking, what is gene therapy, and how to insert new genes in our body. Why? Because this is very related to what we're talking today. That is about membranes. That is, we will talk about liposomes, and there have been a race between two techniques to get genes into our body. One that is liposomes, and that is to have the vesicles. And the RNA vaccines didn't use exactly liposomes; it used another type of technique that is lipid nanoparticles, that is a variation of that. And the other one is to use viruses, that this is how they help these people with this disease. Okay, so when you hear about life hacks, uh, sometimes there are cool ideas, yeah. Sometimes you go hacks, you look for hacks online. There's a lot of crazy ideas that actually don't make any sense. So I don't think that any of us is going to use a roll of toilet paper to hold our phone. Yes, it doesn't make any sense. Or I don't know if this will work. I don't think I will connect my cell phone to an onion to charge it. Yeah. So there are things that are just jokes, or there are things that doesn't work. In terms of biohacking, is a word that it sounds really cool, and it has a problem. Well, Adriana, let's close at least for please change, and then we open it again so it doesn't get too hot. But instead of having this very long article that is very confusing, you say biohacking, it sounds cool. But at the same time, when you look for biohacking, you hear crazy stuff. So maybe you have heard about people using CRISPR. You know CRISPR? Yeah. So I'm going to show you somebody that decided to use CRISPR and inserted So inject a CRISPR to get more muscles. I don't know if you have seen that video. So biohacking it has gained a lot of popularity in terms of having a healthier life, and reach all your potential. And sometimes it could be good things, sometimes it could be crazy things. So one of the craziest things that I have here is about this guy. There is a Netflix show about this guy because about this guy, and John Oliver. I edited this video and he talked about that. So all of you know what is CRISPR? It's an editing it's a gene editing technique that make gene editing super cheaper. Okay, super cheap and super fast. Oh, so this guy said that everybody should CRISPR themselves. Yeah, and he's selling those products, those CRISPRs. There is a 20 minutes explanation of all the things about biohacking, and if you're interested, you can watch a documentary called A Natural Selection in Netflix if you haven't watched it. And yeah, I don't know if the guy actually is making a business, saying to everybody, do you want something? You want to get more muscles? You need to work out. You just have to CRISPR yourself, and he's selling those CRISPR kits. And yeah, so why I'm talking about this, because why this experiment doesn't work. There are so many reasons why it could not work. I don't think that maybe he's not even injecting himself, because maybe it's not smart to inject yourself with the CRISPR stuff. But even if he's trying to do that, inserting DNA, a foreign DNA in our body is really complicated. With fruit flies, if some of you took genetics and I show you several mutations, in our case, those we have like hundreds of embryos and try to inject the DNA, and from those 100, we get a success rate of 2 or 3. Yeah. With humans, you can not say like, okay, from these people that we vaccinated, only 3 got actually immunized. Is so is a very difficult technique, and there are many ways to try to insert the DNA. So now I'm going to show you how a little bit about the the severe immunodeficiency. the door open. Okay, so they even made a movie, and if you see this happen in the 60s, when all this information about the vaccine and how to deliver DNA and RNA in our body happened, they said to me, you need to talk to people about why we're vaccinating. And I'm like, oh my God, this will be impossible to convince some people. And I realized that I need to learn by myself a lot of these things. So one of the cool things about is that with the vaccines that we all had, we were not doing gene therapy, but gene therapy is a very important technique. Yeah, that it could help disease like this one, that one single gene has a problem, we need to repair it. Yeah. So as Felicia told us, okay, so here you see these scientists that are replacing a specific region of DNA. Yeah. So there are diseases that are very complex. Yeah, but there are diseases that are very simple, that you can track a simple gene, and it will be very easy to correct in theory. And to my surprise, everybody was saying, no, this is a new technique, gene therapy. No, nobody has ever tried it before. And to my surprise, we not only were not doing gene therapy with the COVID vaccine, but the first case of gene therapy happened actually in 1990. Yeah, so long time ago. That kid unfortunately died. Yeah, when they opened the bubble, his immune system, I think what happened. Yeah, unfortunately died. But the few decades later, they start, and if you see, it's not a long time ago that they start doing the clinical trials using gene therapy to fix this kind of problems. And you can, if you type this name online, you can see a 40, how old is she right now? 30 something, yeah. Years old person, yeah, and she's still alive. So there is people that have survived thanks to the gene therapy. So that thing that it was impossible in the beginning now is achievable, and gene therapy, this technique that it seems to be impossible, is now there are clinical trials that have solved that problem. And now, if you have seen, you will see in your lifetime a lot of cures using gene therapy. So the key problem, as I said, I show you the biohacking, the guy that wanted to inject. That is really difficult. There are different techniques. One is the liposomes. You remember, to put proteins or to put DNA or RNA here in vesicles. And the other thing that you can do is this: use viruses. Yeah, and these are two competing techniques that we use. Yeah, so the virus is going to go and infect somebody, and we are going to remove the bad part of the virus, and we are just going to infect the people, and the virus is going to inject the good gene and is going to cure the person. Okay, so there are two main kind of vectors, the vehicles that transport the DNA. One, it could be viral, and the other one that is the non-viral, like the liposomes. So let me show you later. I will tell you how now, and this technique has keep improving. We can isolate a healthy gene because we have sequenced it. We take a virus, that is a virus vector. Vector means vehicle. We put inside of the virus. We remove the bad genes of the virus, or we pick a gene that is correct to infect only the blood cells, specifically the stem cells of the immune system that are located in the bone marrow. If you took anatomy and physiology and you suffer with me that class, you remember that all blood cells are the bone marrow. They originate from the bone marrow, and there is where you have the stem cells. You need to infect other cells. You need to infect the stem cells there, and all the blood cells will be now with the healthy gene. And how infecting this now people with this or kids with this disease, babies with this disease, now they will have the right copy. Okay, later I will show you the technique, a doctor explaining that. Okay, okay, we were here. Yeah, I am start pronouncing wrong your name, but thank you. What do you think that it could be the problem of this type of techniques infecting people with viruses? So imagine the virus, you're giving a super amount of virus, yeah, to convert all your body and to replace the bad gene with the good gene. Your body, what it's going to do is going to have an immune response, yeah. Yeah, you can say, oh, I don't know this virus. This virus is trying to attack me. So I'm going to. The bad thing about the guy that he was doing that is that he's putting the field. He's delaying the progress. So later I'm going to show you the first person that died actually in a clinical trial. Somebody already died using with doctors using gene therapy, yeah, in 2010, and this delayed the discoveries for a long time. So this person, this crazy guy in the beginning, is actually delaying that. And the why we don't not use CRISPR or we don't do gene therapy to do all these things, because it's dangerous and because we have to do it in clinical trials, and even in under those circumstances, those techniques can be dangerous in the beginning while we develop them. Okay, so I think that was something pretty cool. And then I'm going to talk more about these things, about, again, these vesicles, these membranes, the thing that we have in the COVID vaccine is all related to membrane, all related to proteins in the membrane, how to take a gene outside of our body and allow it to express it. Okay, and so during the class, I will talk more about that. In the first section of the class, we were talking about that there are different type of proteins in the membrane. Then I started introducing to the topic of how to study proteins of the membrane, and we talk about electrophoresis. Today we're going to talk about different techniques, and I'm going to focus only on 5. And then I will talk about some functions of the membrane. If you want to study for the exam, these will be the topics of the textbook. Okay, so sorry. Okay, so Noella. So last time we were talking about electrophoresis. Do you remember what is the purpose? What is the purpose of doing an electrophoresis? Yes, is to separate macromolecules like DNA, RNA, and in this case, proteins. Yeah, and you use, Jenna, do you remember what are the parameters that you use to separate them? Remember how we separate them? Electrical charge, very good. And we use also, if you see here, what I tried to show in at the bottom, here is the DNA. Yeah, and here if we had a microscope, you see this membrane, so you're also, it's not only by charge, but also the length, because the small molecules will definitely cross much easier than the long molecules. Okay, so, oh, sorry, it's an old version of the presentation. So this should be analysis of the proteins. This technique separates molecules based on the size. We use charge and size, and we separate the electrophoresis. We separate DNA, RNA, and proteins. And page, sorry, I think you will never forget this technique. There's one specific about one electrophoresis that is specific for proteins, and is SDH page. Yeah, and it's mainly to separate proteins. Okay, so with an electrophoresis, so next time that you study with John and he asks you about electrophoresis, when you take the methods, you need to remember that there is a ladder that is very important. So last time Duncan told us about the electrophoresis gel. You need a way to visualize how the proteins are moving. So you will have this is like a scale bar of the map of Google Maps that you say, oh, this is 1 kilometer. Yeah. So you know it's okay, it's 80 kilometers. I will not be able to walk this. I will need to take a bus or airplane or something like that. Here it can tell you how big is the DNA or the proteins. Yeah. So so the electrophoresis is the ladder is a molecular weight marker of a size that is a standard, and they sell it for you. If you see these have different colors, the proteins are the DNA. They don't have any color, so you need a dyed. Yeah, and the dye should be for DNA or RNA will be Jill uploading purple, and for proteins will be bromophenol. Okay, so two things that are important when you're doing an electrophoresis, to have a dyed so you can see how the proteins move, and to have a ladder to interpret the data. So here, this is an example of a ladder in a SDH page. So here you can see that they are in kilodaltons. Yeah, why? Because this is a measurement of weight. Yeah, for proteins. So here you can see the proteins that have been run. And so remember, the kilodaltons is a measure for proteins, and here I explain what a kilodalton is. Okay. So now that you know that there is a dyed, that there is a ladder, I'm going to show you an example. This is an schematic of what you have, and here is what you will see in a real sample. So here you see that in kilodaltons, you will have from the larger to the smaller. So the smaller one is going to move faster. Yeah. Here you will have the ladder that it can tell you that this is 250. This one here, it will be 150, and the smallest one here is 20. If you remember last time that here you have the sample A and sample B. And Duncan last time, he told us that when he did it, he pipette the DNA or the proteins or the thing that he pipette, he put it in a hole. Yeah, and that hole is called a well. That is just, imagine that you have a fork in yellow, and you put it's actually very similar. So you make holes there, and in those holes you can put the DNA that is going to move. So you put in the holes here. You when you make the gel, you use like a fork there, and it will have those very thin wells, and in those wells you are going to pipette and put the sample, and the sample is going to move. Eric, what is the size of this protein or this DNA or protein? Yeah, what do you think? What is the size of that protein? So here you have your ladder: 20, 25, 30, 50. This one, it should be between
37, 50.
Yeah,
So 45.
good enough. So if it appear in the test, you know that if the answer is 300, 2000 oxygen, you will say, no, it will be 40K. Yeah, so pretty easy to understand this. Maddie, the third one, what is the size of this band, the number 3? Yes, good. And okay, and this, do you see that there is this large band? here, that band, Duncan, I don't think that you remember that. Do you know why we have such a long band? Why do you see it is not in one, is continuous in all of them, and it's at the end? Why do you think that we have that band?
That, like, against.
To say stop now, because if not, all the sample is going to yeah. So here you say like this is the end of the sample. If you can continue running this forever, and all the proteins are going to go outside, but so this you have a dying front. That is, it tells you here is where you have to stop, and you stop the machine, and now you can take the sample. Okay, so I will be very happy when you go and John will ask you, and you say like, yes, we know this, because he will ask you. And if you take the materials and methods with him, you will be doing a lot of these techniques. Okay, so what is not, last time I asked you what is not an electrophoresis. This is a gel used for in molecular biology. So we use it to separate proteins based on their size, to separate DNA fragments, to then determine the sequence of RNA molecules. Why? Because first we separate it by size, and then we will use another technique. But definitely the amount of lipids in a sample is not, we don't do it. We are not use electrophoresis for that. Okay, so now, Paige, your question. What is the purpose of this? So look, the page come from this, here is the P, the poly and the A come from here, and the gel, here's the G and E come from here. So this is the material of the gel. Could you guess or not? Could you guess, help us here? Think that you're right, but I don't trust myself right now at this hour. So here we have this SDS here. That this is the long name. Yeah, that is a chemical that what is going to do is the proteins could be globular. What is going to help you to do it is make it linear. Yeah. So when you put it in this gel, the proteins will move easier because it will help it to move, to make it linear. So it will help you to solubilize the submembrane fragments and separate proteins from another one, and it's a technique to use from proteins and protein-lipid interactions. And here, so here it shows how it works. So here you have these proteins that are folded here with negative charges. And if you remember your chemistry, when you took this long chemistry, many hours of chemistry with John Brubaker in the beginning of Cells and Energy, you remember that there are many bonds here, and here these bonds are very important. Those are broken by this chemical, yeah, that we put here. Here is going to reduce it, and is going to allow to unfold these proteins. And here you have also the SDS is going to produce negative charge, and so we're going to go from folding proteins to linear proteins that it can be analyzed here. Rico, do you remember what is the name of these blue bands? Yes, do you remember? That is called the ladder, okay? Ladder, yeah? So it will give you the molecular weight, and here it can give you it will help you to read the sample size. And here, for example, in these two samples, you can have two proteins, so you might have problems, yeah? Because you have proteins that are the same size, but here we just know that they have the same size, but we don't know the sequence. This can be actin, and it could be also myosin if they have the same size. So we need to have an extra technique use another technique to actually separate between myosin and actin, or coherent and another protein, or protein 1 and protein 2. Yeah. So the next technique that we will see is in the next section that it will solve that. It will be the Western blot. Okay, so we will talk about that in a second. Here is all the information that you need to know. I'm sure that you remember it, but in case that you have any question, all the information is there. And page, very good. To separate proteins based on size. Okay, so and this, the end of the class, we talk about at the end of the previous class, and in this beginning, we talk about how to work with proteins that are located in the membrane, some of them that are transmembrane, and some of them that are in the periphery, or some of them that are anchored. And another important technique to separate those proteins is to use this electrophoresis, and it's very important, specialized for proteins, is this SDase page, that it make the proteins linear, and then you can remember, use the ladder, yeah, to separate, to calculate the distance, and then with the Western you can differentiate between, to see if your sample has a specific protein. Okay, so we finished that section. Sorry, about the isolation and analysis of membrane proteins. Now we're going to talk about other techniques. And here I'm just going to focus on 5, but this can take, you can be all the semester. Actually, the materials and methods is just focus on techniques. There are so many. So, Asher, what do you think immunofluorescence means? Good. Onto what? Your fluorescent, perfect. What is the first word? Into antibodies. Very good. Yeah. So you use an antibody that it will find something and attach. Yeah. And you put a fluorescent. So very good. The Western blot, I already told you, Angus, more or less a little bit. So you use, again, this complementarity to differentiate between 2 proteins. We can use liposomes too. And I already gave you a function of the liposomes. In this case, you can just, you have a doubt about, okay, where I isolate this protein, what is the function? We can put it in a liposome and study the function there. So we have immunofluorescence, Western blot, membrane reconstruction, X-ray crystallography, that is what you do with the DNA. Yeah. This to take a picture to infer this atomic model, and this word that maybe I saw that some of you write in the summary, that it seems like something very weird and impossible to understand, and it's actually very easy. So Paige, no, I already asked you too many questions. Julia, pretty easy question. What is hydro in Greek? Perfect. This is a little bit more difficult. Pathy. I didn't know. Don't worry. But it sounds like pathology, that it could be suffering in this case. Yeah, feeling in this case is interaction. So we're talking about interaction with water. Yeah. So here you're going to see which proteins are interacting with water and which proteins are not interacting with water. Yeah. And with that, you can see which proteins are in the membrane and which proteins are transmembrane sections and which are not. So 5 techniques, and I'm going to tell you about that. So I already told you that one of the key techniques is use affinity label. So if you see all these guys, they don't attach here. Yeah, there's only one that it attached perfectly. Yeah, that it will be this one, because they are complementary. So I don't know if you remember the lock and key metaphor that you use when we talk about enzyme and substrate. Yeah, you have to have a specific molecule to do these kind of things. Yeah, in the same way that you need to have a specific key that have the specific shape. So same thing happened with enzyme substrate. Same thing happened with receptor ligand that we will talk about later. That is how you send signals. And the same thing happened with your immune system. The antibody and the antigen, again, the lock and key. So they just have to have the same thing. And in addition to that, you add a fluorescent molecule, a radioactive molecule to allow you to see the protein, okay? So again, we already talked about all this fluorescent stuff before, and here they are showing you a picture that you have green and blue. I don't think that you can see here, but here if you want to see all this tubulin, so you have the protein of interest that it will be tubulin. So you're going to have the lock and key metaphor, you know, that they have to attach very well. They have to be complementary. So you're going to have an antibody that is complementary, that it will be the anti-tubulin. Yeah. Then you will have, this is the primary antibody. Then you're going to have a secondary antibody that is going to attach to the primary antibody. Again, it will be just complementary, and this will come attached with a fluorophore. It could be a GFP. It will be with something that is fluorescent. So in other words, we are just reusing something that the cell has been using all the time. When you learn in cells and energy, enzymes and substrate, and also in chemistry, this course you're going to learn about signaling, that are receptors and ligands, and also about antibody and antigen. All of them are just complementary. We are just using that and putting something that is fluorescent or that is radioactive to be able to see it, okay? Oh. Okay, so let me play the video, sorry. Oh, sorry. Last time. Okay, so I'm going to pause the video so we don't spend all the time watching this. See your nap. But the idea is here we can see where is the protein located. And it's in a, yes, Arnab is Arnab, yes. So he no. The proteins are located in a specific place at a specific time, and you can locate it. Yeah. So the immunofluorescence is a very powerful technique, and you will hear about that everywhere. Okay, sorry. The other technique that we are going to use is a combination of all of them. That is the Western blot. And in the Western blot, the first thing that you do here, if you see here, is the electrophoresis gel. You see it here. And now as I told you before, you can have protein that have the same size, so you need to separate them. So we use this complementary lock and key technique, and we are going to transfer those proteins because we're going to have a membrane that is going to attach those proteins. Okay, and then we are going to stain it using the same kind of technique. So you're going to have something that is fluorescent to see it. So in other words, we separate it, then we put a membrane with the protein that we want, and then we also have a fluorescent molecule. And as a result, you're going to end up seeing only the protein of interest. So for example, if it was tubulin, and tubulin is here, you're going to have a membrane that has an antibody that is anti-tubulin, then it will have a secondary antibody with a signal that it will make it fluorescent, and you will visualize it like this. Again, you will see you will have the ladder here and the different cells that you study, and you can test that the protein is there. Okay, so again, this is one of the protein techniques that you will hear forever, Western blot, and it'll help you to detect and identify the individual polypeptides. So it starts using electrophoresis for the separation, and then you need to transfer the protein. So you start with the SDA page. Here you will find the gel and the results. Then you need to pick, in this case, you have 3 examples of proteins and you want to decide which one is the correct one. So you need to make a Western blot, okay? To decide to see if it's the green one, the blue one, or the purple one. The other technique is the liposomes, very important technique that I already told you that is making these vesicles, and you can put proteins inside of that. And later, and this is important to study, the function of the cells, of the membranes, of the proteins of the membrane, and then I will tell you about a variation of that. The other technique that we have talked about is the X-rays, the X-ray crystallography. So here you have a crystal, yeah, so you can take, for example, to find the double helix shape, what they did is take the DNA, crystallize it using some salts, yeah, then to put they took X-rays, yeah, and as a result of that, they were able to see the diffraction patterns. Yeah, so here you use an algorithm, the Fourier transformation, and this will tell you the electron density map. Yeah, so you go from a crystal, then you can start getting some indication where all the atoms located, and then you can create a model of how this protein or this DNA the atomic model of that. Yeah, so using just this simple technique, we have been able to find the shapes of many proteins. The first one was this one, in Pseudomonas, and more than 500 protein structures are already found. And again, you already know about this because of this is the same thing that we did for the DNA. Okay, Angus, sorry. So could you read it? And
How are amino acids typically represented? Dendropath.
okay, so weird stuff.
Their size.
Okay, we will see. Okay, so remember what Julia told you: hydro, water. That make more sense, and it will be pretty easy. So here you see the graph, and here we have the index of the hydropathy, and here you see the amino acids. Yeah, so you see that there is one region that is hydrophobic and the other one that is hydrophilic. Yeah, so it can help you to locate the three-dimensional structure of the membranes. And why? Let me see. Here, you see that there are sections that are in the periphery, and there are some sections that are in the membrane. Angus, these ones that are in purple will be hydrophobic or hydrophilic. This one in purple. Hydrophilic and hydrophobic. And look.
Andrew Paul.
Yeah, so the ones that are outside here, they will be hydrophilic, and the ones that are in blue that are inside of the lipid will be hydrophobic. They don't like water, so they will move there. Okay, so this is how it works. So you will measure these two things, the hydrophilicity and hydrophobicity. Sorry for those weird terms. So here you have the membrane topology. And as you can see, this is what we were talking to with Angus. You have these represent to the blue area, the one that are the transmembrane that don't like, that are located in the lipids. As they are located in the lipids, they don't like water. And on the other hand, you have the purple region. That is the one that like water. This is why they are outside. And here you can see again the same thing. So here you see in blue the protein, yeah, the membrane topology. So you see this one is here. So it will be inside of the blue region of the, so this part will be inside of the, close to the phospholipids. The same thing second, third, and fourth. While this region of the beginning, it will be the one that is hydrophilic. It will be actually in the cytosol. Okay, the slides are there. And why this is important, because it helps you to create the three-dimensional structure, and it can help you to understand the protein folding, the stability, and the interactions of a protein. So we don't have a lot of time, but I will post the video in case that you want to watch it. Angus, good. So how are amino acids typically represented by this hydropathy plot? Because they tell you the hydrophobicity and and if they are hydrophilic. Okay, so I told you about these 5 techniques. So Angus no. Asher told us, just by the name immunofluorescent, you have immuno is antibody, a protein that is going to attach to anything with a fluorescent marker. Then I told you that the Western blot, it allows you to differentiate which one of these bands. You can have bands that are very similar in size, but you can say with the Western blot, you can say is actually acting or is actually coherent, because you are using this complementarity that you can see here. You can watch, I think there was a video that I posted there in case that you have doubts about the Western blot. We already talked about the liposomes. You already know about the crystallography, and maybe something new is this hydropathy, but I'm sure that now you understand much better. So this is the guy. You can find the article of this guy. There was actually 2010. There is a Nature article talking about the clinical trials of this guy. It sent the field really long, like nobody want to invest anymore in gene therapy. It took like 10 years to recover. And this is why it's so important to talk about that. If you read about the article about this guy, he actually was not going to die about the disease. He was heterozygous for that disease, so he could have a normal life if he had a balanced diet, if he had a metabolic disorder. But he wanted to help science, and he enlisted in that clinical trial, and unfortunately, too much virus, too strong immune response, and he ended up dying, and it was very sad. So this is how generally happen when the immune response. You have a virus, so the body goes and said, we need to kill this virus, and then you, as a result of that, you create antibodies that it will attach to the virus in the same way that the Western, the antibody staining, all the things that immunofluorescent, all those things, and there will be cells that will have those memory cells will have the antibody. So every time that a virus comes, it will be okay. Felicia, Julia, what do you think that is these lipid nanoparticles? What are lipid nanoparticles used? They were the ones that we use in the vaccines. I think so. I think so. Let's. We are taking the tests. One thing as I put now the answers cover. Sometimes I'm like I don't remember many of the things that happened, but sorry, I know I'm too tired. But I think this is we will see in a second. So this is all the things that happen when you have a vaccine and all the things that you have to check. But I wanted to show you this. This is a liposome. Do you see that, Felicia? What do you see? There is the difference between a liposome and a lipid nanoparticle. Not just from the figure. What do you see? Which one of those is a bilipid, bilayer of lipids? The liposome. So the lipid nanoparticles. And look, so here you can have. Do you see these represent a hydrophilic drug, so where you can put it. And here you have micellus, yeah. So you have specific regions that you can cover there inside. And here you can have a specific RNA or a specific oligonucleotide, okay? So those looks very similar, but they are very important. And the one that we use for the vaccine, it was this one, okay? So they are specialized to put RNA and then DNA molecules, the lipid nanoparticles. This one can be used for other things. This one are specialized for RNA and DNA. So as you can see, the guy that put the DNA and inject the DNA, that is a very complex and complicated process. It's not as simple as taking a kit. So we have vectors, like one of the first kit that we saw, that are viruses, and we can have liposomes, or we can have lipid nanoparticles. Sorry, this is just really quick. Okay, you can see this. So here they will talk a little bit. Okay, sorry. So the answer, Julia, I'm going to play a video later, but the answer, it was to target drug delivery. Yeah. So these lipid nanoparticles are especially good to put any type of DNA or RNA. One thing that I didn't know before the vaccine and all these things is that in the same way that we have an evolution of many structures or many tools like a pen, that we start with feathers, and this is why we write in cursive. Sometimes that because you don't want to put pieces of ink, so this is why you don't try to lift your when you're writing, but we have techniques that have changed and modernized and make it so easy that right now nobody's going to complain, like a little bit if somebody steal a pen from somebody else, but nobody's like, oh my God, thousands of dollars. The same thing happened with vaccines. We have changed a lot the ways that we have worked, and we have gone through. I don't know if you remember. So from 2,000 years ago or longer, I don't know how many years ago. Do you remember that there was a report that in China people was infected themselves with a disease that it was in cows. Why? Because the sister virus, a virus that it was related that actually killed the humans. If you infected before with the virus that affect the cows, later when you get sick with the other one, you didn't get sick. Did happen many thousands of years ago. Nobody knew why. Now we know why. Why? Because you have a weakened virus, yeah, an attenuated virus, you got sick, your body produced antibody. When you got the virus that actually infect humans, you didn't got sick. Then we understood the science. Then we got inactivated virus. We removed the DNA or the RNA. So we just have the proteins that we have been talking about, protein membranes. And this is the other generation of virus. Then we use subunits of the proteins, yeah. And we can also have viral vectors to have the virus. Now we have another one that is to put lipid nanoparticles. Why we don't do all these things? The same reason why we don't use this anymore. Because it's much more expensive, take longer time. We don't imagine taking notes, all of you, like it will take forever, yeah? Same thing happened with the lipid nanoparticles. It will help. It will take a long time. One of the key things that I didn't know is that I thought that technique emerged long time, like really recent. It actually, the lipid nanoparticles have been using since 1970. And if you watch the show Narcos, very famous guy, scary guy. But what you're doing with the antibodies, all those things that we were doing, in all these type of techniques, we were just doing biological mock shots. So if you ask a Colombian what this means, this is a picture where the first time that they caught him, and it's not a joke, he was laughing because he knew that nothing, he was so rich that nothing was going to happen to him. But when they found that picture 20 years later, they sent it to the Interpol, to the American embassy, and he was caught because then everybody came to Colombia. So what happened is that the virus, what we do with a vaccine is doing exactly the same thing. We take these antibodies, and the body is ready to receive to get any disease. So antibodies are just biological mock shots, and you can do it with viruses that are dead, with viruses that are weakened, with pieces of virus, or you just need to have a lipid. No, sorry, you just need to have the RNA of the protein, of the spike protein in the case of the coronavirus. And why you make it like that? Because it's much cheaper and easier. You don't need to create the protein. The ribosome is going to fold it, translate it and fold it for you later. Very quick. Sorry for taking so long. This is a long video that you can watch. It's a long video, but again, if I tell you, like, I couldn't, when I was talking to people that they were anti-vaxxers, it was so difficult. And I said, okay, so in order for me to understand that they understand me, they will have to take cells and energy, genetic revolution, maybe genetics, maybe cell biology, all these things, and maybe that course with John in techniques in molecular techniques, and I was not efficient because I didn't know also this information. I know that, for example, if you see this, for me, it's fascinating to think that this is not something new. If you remember the video, they start working this in 1978, and as I told you, we know that the basis of the immunization just by tradition thousands of years ago, and what we are just doing is make it simpler in the same way that we are making pens simpler to use, or a laptop. Every time that there is a new version of a laptop, it's not something completely new. Yeah, it's just a small modification, and we just didn't know. And I think this is a problem actually of the government. Government now not only need to spend money on science, but also in communicating science to people, because we don't know. I didn't know all these things. Okay. Sorry for taking too long. 5 more minutes. We take the quiz. No more cell biology until tomorrow. And so we were Felicia. Sorry. Do you remember any of these proteins and stuff, cells and energy? You remember what is the function of this electron transport change? You remember what they were doing? More or less. I know when every time that I have to teach that, the aquaporins. Shia, do you remember what are the function of the aquaporins? So aqua, water. So they are just porins is like ports, so they bring water. Yeah, the carrier proteins. You remember those carrier proteins, Noella, what they are doing? And finally, Jenna, you have seen this. Actually, this is a video that is the voltage gated channels. So they are bringing sodium and potassium and all those things. All those proteins are involved in the membrane. So there is so many functions. The electrotransport change, the mitochondria and the chloroplast, all of them have functions. You have transport proteins that, like the carrier protein that Noella was talking about. You can also have channels like Jenna was telling us that they were very important, the sodium, potassium. And we have also ATPases that are the ones that produce the ATP. So those are a lot of proteins, and we have even more. So very quick, we have proteins for the endocytosis and exocytosis. We are going to study those. And again, I apologize from the beginning. You will be overwhelmed with all the names. I'm not going to ask you all the names, but you will see that we went a little bit crazy to understand every single protein, how they are able to do the endocytosis. And this is why the nanoparticles, the lipid nanoparticles are able to do endocytosis and break, and you have the messenger RNA inside of the cytoplasm, and in an efficient way, because we studied that. Same thing with exocytosis. Maddie, what is phagocytosis? This is not exactly phagocytosis, but more or less. Yes. Yes. So I think it's autophagy. Sorry, autophagy, self-eating. So actually, the mitochondria is not going to go and run away from the cell. We are going to use lysosome and hydrolysis, and they are going to break those. So very important phenomena, cellular phenomena, and the person who discovered it, he got a Nobel Prize. They are also receptors, and again, Julia is going to talk about in her poster about that. This is going to be a long class when we talk about signaling, there is so much information about that. And this is something new. You have heard about neurotransmitters and hormones in our classes. Here, this will be part of the biosignaling. And this is the cytoskeleton. We give an introduction to that. We are also going to talk about that. We also know that the proteins can have different orientations. Some of them can be outside and some can be inside. So the ones that are outside will be receptors generally. This one will bring materials inside, and this one will be the one that transfer the signal to the cell. And to finish, where we are, Rico, there is the protein generally, and lipids generally have sugars. There are two types. Do you see what is the difference between this one and this one? There are two types there. In the arrow, you can see the difference. Can you tell me what is the difference there? One is linked to the amino acid, yeah, in the nitrogen, yeah, in the nitrogen, and here is the carbohydrate. This is why they are they have this carbon. The proteins will have a carbohydrate, and here do you see that instead of nitrogen, it will be the oxygen. So when you have membrane proteins and you add a sugar, there are two types, one that attach in the nitrogen and the other one that will attach in the oxygen component. And here is all the information of the glycosylation, that is the addition of the carbohydrates. And this is what just Rico told us about the N link. And in the next slide is the O link. Okay. In the next class, I will talk about the other few slides. You can start taking the quiz now if you haven't taken it. And that's all for today. You can leave when you are done with the quiz. If there is any typo, please let me know and I correct it.